My name is Tommy Das. I am a biology major at Dartmouth College, and I'm writing my thesis on the effect of oxidative damage on meiotic chromosome segregation in Drosophila. So meiosis is the process by which we make these cells that are going to carry on our genetic lineage, so sperm and eggs. What it is essentially is two sets of cell divisions, and in those cell divisions, one of the big points is to make sure you get the right amount of genetic material, the right number of chromosomes. During meiosis 1, your chromosomes are going to pair up and then segregate to opposite poles, so they're going to be in two different daughter cells. What maintains that the homologs separate to the opposite poles is cohesion that exists between the arms of the homolog sisters. If that cohesion is lost, then chromosomes can segregate to the same pole, which will result in cells that have too many chromosomes. And if these cells get fertilized, then you're going to have developmental problems, or most of the time those embryos won't come to term at all. And my project is dealing with the effect of oxidative damage on defects in meiosis and non-disjunction. As we get older, we're more prone to having these non-disjunction events, and people think that that's due to cohesion being lost between the arms of the sister chromatids. It's also been linked that as we get older, we have an accumulation of what are called reactive oxidative species, which essentially are oxygens that are missing an electron and are a natural byproduct of the mitochondrial electron transport chain. So because these oxygens are missing an electron, they're chemically reactive and can pull electrons off of proteins and subsequently damage them. So we've tried to link these two things together and said that maybe why we're losing cohesion with age and why we see an increase in non-disjunction with age is that we have an accumulation of reactive oxidative species that is damaging the cohesion and causing us to see more non-disjunction with age. If we increase the amount of oxidative damage in our flies, are we going to see an increase in the amount of non-disjunction? So by using molecular biology techniques, we've been able to knock down the protein responsible for cleaning up oxidative species, and so consequently we've increased the amount of oxidative damage. We take the flies that have the increased oxidative damage and cross them to one of our lab stocks of flies, and based on what the eye shape of the progeny are, we can actually see if the parent flies had more gametes that have the incorrect number of chromosomes as opposed to correct number of chromosomes, and that way we can get a number and a readout for non-disjunction. And in flies that we've increased the amount of oxidative damage, we actually do see a small increase in non-disjunction that is statistically significant. This increase is a lot smaller than we were expecting. So and part of the reason behind that may possibly be that there's a system that's responsible for properly segregating chromosomes even if they've lost cohesion. So this system, called the achiasmic chromosome system, is going to actually ensure that you still get the right amount of chromosomes in your gametes. So if we knock out and damage that system by using a mutation in a critical protein for that system, we're actually end up seeing a significant and much larger increase in non-disjunction if we couple that with more oxidative damage. Essentially, this data shows that if you increase the amount of reactive oxidative species and oxidative damage, we're going to see more defects in terms of meiotic segregation and non-disjunction. However, we're not quite sure exactly what the target of these reactive oxidative species are. It could be the proteins that are involved in the cohesion between the homologs, or it could be something else involved in the segregation pathway. So what we'd like to do, ideally, is find a way to pinpoint that target and do assays to see if it is cohesion that's being lost and if these crossovers are happening and then being lost due to oxidative damage. So ideally that's where we'd like to take the project in terms of seeing if that's what the reactive oxidative species are targeting. If we do show that link between reactive oxidative damage with age and increased non-disjunction with age, we could incorporate that in some sort of treatment where we're treating for oxidative damage, trying to find a way to decrease oxidative damage through some sort of intense antioxidant treatment. Maybe that could possibly decrease an older woman's risk of having a child with Down syndrome or having a child that comes from an aneuploid gamete that doesn't come to term.